So while we're working on that, my name is Marc Tewey. I'm the CEO of a company called Biotheraputics, which is a startup biotechnology company which licensed the PKU probiotic from Catherine Deming's group at the University of Texas, which was funded by the PKU Alliance. So we'll wait on our slides here for just a minute. There we go. Okay. So Trayer is basically as I mentioned, so it's a startup company. It was a company that was basically formed around this probiotic project. We're a group of 5 people. We are basically rare disease drug development and commercialization people, and we also have a tremendous amount of expertise in probiotics. So my personal background is I've been working in rare diseases at a company called Sigma Tau for most of my career, and I'm not a scientist. But I'm a person who helps commercialize these medicines and then move them forward into clinical trials. I also had an opportunity to launch a probiotic many years ago called VSL3, which is a probiotic that became a very, very commonly used probiotic in the gastrointestinal field. And so when I saw this project, I immediately said, oh my gosh, this is something that I really have to be involved with. I also had an opportunity to work with David and Lynn Powella at Cambric Therapeutics for a number of years. So to me, this was PKU, it was rare disease, it was probiotics, it was all the things that I really love. So I had to get involved with this project. So I was very happy to be able to license the technology from the university. So what we did is we formed a team. As I mentioned, we have a manufacturing probiotic expert. We have some clinical development experts, and then we also have one of the godfathers of the field of probiotics, a guy named Claudio Di Simone, Professor Claudio Di Simone, which was one of the gentlemen who many, many years ago was really working to push the field of probiotics into the gastrointestinal field. So we're very, very happy with this team that we have. The team is very important. Any biotechnology drug like this, it needs a brilliant researcher like Catherine Deming to invent this thing and get it established, and then it needs a team of crazy people like us at Trayer who are willing to quit our otherwise normal jobs and go dedicate time, money, effort to moving these projects forward. So our job is to get it manufactured, figure out how do we make this stuff in a real production factory. How do we get it in front of the FDA? What's the regulatory path? And then the big effort, the big job of us is to ultimately raise money to move it into its various phases of clinical development. Money can come from a lot of sources. Some money has come from the PKU Alliance already. They're government sources. But then also one of our big jobs is we're going to get this in front of venture capital funding to then begin to move it into clinical trials. Projects like this cost tons and tons of money, and they're very, very risky. So you need to have people out there who are willing to bet huge amounts of money on these projects in the hope that maybe they're successful one day. So it's a real enterprise. So let me talk, first of all, before I get into the project, I'm going to talk just a little bit about the human microbiome. This is something that's come up several times over the last 2 days. I'm glad to see this come up. This is something it's really a new field of medicine in a lot of ways. It's been driven by a lot of science that's gone on in the last few years. The NIH has a huge project called the Human Microbiome project, which is seeking to sort of characterize and map the whole human microbiome. Basically, we are all just walking condominiums with bacteria. We are filled with literally trillions and trillions of bacteria. They live in our GI tract, they live on our body, and most of us consider bacteria to be bad stuff, right? They're not all bad stuff. Actually, a lot of the bacteria that live in and on our body are good stuff. They're our friends. We need them. We were born sterile. We acquired bacteria from mom and dad from the very, very first moments of our life, and we evolved with those bacteria. They evolved with us, and they play a variety of different important physiological processes in our body. They help us modulate our immune system. They help us break down foods. They help produce anti-inflammatory compounds. They help us make vitamins that we can't make ourselves. And we are really just now beginning to understand the full implications of the human microbiome on the body. Lots and lots of different species of bacteria in our body, and we're still trying to understand all of this. There's neurological implications that are tied to the human microbiome. All kinds of stuff is going on. We're really just beginning to understand it. This is, by the way, why the work that Michael Allen is doing is so important. We really need to fully characterize the microbiome of the PKU population, which we know is different than the otherwise non-PKU population, but it's very important to understand that because there may be things that we can learn there that could help us develop new medicines or maybe change the way that we use current medicines. So we already know we can use bacteria, good bacteria, probiotics, to affect certain diseases, right? 15 years ago, this was a very, very new concept. This concept of eating bacteria for positive health benefit. In 2003, I launched this very commonly used probiotic called VSL3, and I can remember going to doctors and they would say, Americans will never eat bacteria. You're crazy. Well, the data overcame the sort of that apprehension. And so over time, over the last really 10 years, we've accumulated a tremendous amount of data on probiotics in the field of inflammatory bowel disease, IBS, pouchitis. So there's a lot of probiotics out there that are commonly used. And forgive the selfless promotion, but the probiotic there called Visbiome is my probiotic. It's a very high potency probiotic, one of the most well studied on the market. That's used in inflammatory bowel disease. But we're also studying it right now in two very, very exciting studies in patients with HIV. So one is a multi-center NIH-funded study with the AIDS Clinical Trial Group. Patients with HIV have a very, very different gut microbiome compared to normal, healthy people. We believe that it's not a healthy gut microbiome because of their disease, and as a result, certain bacteria are just flowing into their body, and it's causing a lot of downstream comorbidities associated with the disease. So even though we've effectively have good treatments now for HIV, there's all sorts of comorbidities and other things that are going on with this patient population. We believe that we have a good shot at helping them with this product called Visbiome. So anyway, it's some exciting stuff that's going on. So what about probiotics as drugs? That's what we're talking about doing with this project. So this project is, we call this recombinant L-acidophilus SA412. That's the official name of this. So the concept of using probiotics as drugs is really kind of new. One of the first of these probiotics to get in front of the FDA was a project I had an opportunity to work on at Sigma Tau, which was for a disease called necrotizing enterocolitis. But what's happening here is that the FDA is beginning to accept this idea of using probiotics as actual drug products. So they've issued guidance, they've given a lot of direction, and now we have a number of companies that are out there publicly acknowledging that they are working on probiotics as drug products. One of them is myself. Synthetic Biologic is one. Cirrus, Synlogic, who's here, I believe. What's really, really exciting for the PKU community is that of all of there's about 12 companies or so that are working on live biotherapeutic drug products, and 3 of them are working in the field of PKU on very similar targets. So as a patient community, you should be very pleased about this. So basically you have now 3 companies with a lot of brain power, all focused on PKU. And it's been now identified that this is a target for drug development. So this idea of modifying a bacterium, consuming it, and then having it help us catabolize the phenylalanine that's in the GI tract. Okay. So anyway, this is I think very exciting. It's also good to have, from a PKU community perspective, it's good to have this competition. You want a lot of companies out there trying to make drugs for your patient population, and that's what's really going on here. We see this a lot in drug development, where a pathway will be identified scientifically, and then you'll have a number of different groups that will go and try and develop drugs based on that same kind of pathway. We saw this in Pompei's disease. Anybody see the movie Desperate Measures? Did anybody see that? A Harrison Ford movie. So that was a story about this guy, John Crowley, who had 2 kids with Pompei's disease, and he started a company that tried to create an enzyme replacement therapy for Pompei's disease, and simultaneously there was 3 or 4 other companies trying to do the same thing. There's a book named The Cure, which is a better book if you're interested in drug development, about this whole story. But what happened there is these multiple teams were all working at this same sort of target, and ultimately they actually got together and formed what became Genzyme. And then they sort of took the best pieces from this guy's project and that project, and ultimately that's how the drug moved forward. So who knows, maybe a similar kind of thing could happen with us. So why is it that there are these probiotics that are becoming developed as drugs? Well, it's a lot of things. It's, first of all, the FDA has given us much better guidance for how to do this. These bacterium are much better. There's just been a huge number of technological advances that enable us to better characterize these bacteria, better identify their genomic structure. There's now better manufacturing technology that's available to make these bacterium. It's really super hard to make these bacteria in a biologic environment because you essentially have to make a sterile product, more or less, except for that one thing that's living, that you want to have living, this probiotic that you're going to consume. So when you think about that, that's sort of complicated to do from a manufacturing standpoint. So also there's a lot of target indications outside of PKU that have been identified in inflammatory bowel disease and other things. There's a lot of activity going on in this field, a lot of excitement in this field of the microbiome and developing drugs around it. Okay, so let's talk about recombinant Lactobacillus acidophilus SA412. So if you saw Catherine Deming's presentation yesterday, so this is a genetically modified strain of Lactobacillus acidophilus, which has been developed to express this enzyme that is otherwise missing in people with PKU. All right. So in theory, we'll be able to consume this, and then this will help us to catabolize or sort of eat the phenylalanine that's available in the diet. So the goal would be to use this as either an adjunct to the medical food diet or along with other drugs to reduce phenylalanine levels in patients with PKU. And we were happy to license this from the University of North Texas, and all the original funding for this came from the PKU Alliance. So how do we make this? Well, this is just the simple non-scientist's presentation on this. We basically take a yogurt bacteria, right? This is a yogurt bacteria which has been in the food supply for almost 40 years. It may be in that yogurt that's back there, actually. It's called Lactobacillus acidophilus, and it's a particular strain that's been shown to be very, very robust in a manufacturing environment. So it's been very stable. This is extremely important from a drug development perspective because we can't concoct a bacteria that eats phenylalanine but then have it die in the petri dish a week after you make it, right? So it needs to be stable enough that it can get into the drug supply so then it can be used by people. So this is a very, very hardy strain of Lactobacillus acidophilus. It is the genus and species, so Lactobacillus acidophilus as a class is a natural resident of the human GI tract, so you will find this in a lot of us naturally. And so what we're doing with this strain is now we're taking it and we're modifying it by inserting this chromosome with this AVPAL gene, now making it capable of chewing up phenylalanine. So the theory is that the acidophilus is sort of at home in the GI tract. This is where he lives, right? So he wants to be there. But the issue with this bacterium is he's unable to make his own phe, right? So he's going to want to try and acquire phenylalanine from his environment, and then what he's going to do is he's been engineered to internally produce the enzyme that can catabolize the phe. So he's going to kind of go out into the environment and hopefully grab some of the phe from his environment, and he actually catabolizes it or sort of neutralizes it internally. Okay. So that's the theory of the technology. We'll see. And as Catherine mentioned yesterday, it's been shown to work in animal models, but we still have lots of work to do. So where are we right now in terms of development? So as Catherine mentioned yesterday, we've proven this technology on a strain of bacterium which was not well suited for long-term human development programs. So now what we're doing is we're taking the modification and moving it over to the L. acidophilus strain. So we're in the process of doing that right now. Once we're completed with that, we will do in vitro tests, so sort of petri dish tests, and then we'll also do several animal tests to see if we can reproduce this action. From there, we will move to engaging with the FDA to begin to put together a regulatory pathway for this. This is a bit of a unique project, so we need to engage with them fairly early, but you need to have that sort of basic animal set of data before you do that. And then really the next important phase is the manufacturing phase. So it's very, very, the leap from making a product like this in a bench in a lab versus making it on large-scale production and then lyophilizing it, making it stable, that's a big technological leap that lots of drugs fail at, unfortunately. So we have a lot of work to do to get that to that next phase. All right, but this is where our sort of expertise and our manufacturing team expertise comes in. And then there's some additional non-clinical development we'll have to do before we can start to entertain human clinical trials. So the goal would be to establish that we can manufacture this, get the gene insertion done, and then engage with the FDA and receive permission to engage in the first human studies by 2017. Now, whether the study would actually start in 2017, that's probably pretty optimistic. But that's the goal, is to get the regulatory clarity so that we can begin to prepare for the clinical trial. So how could this product ultimately be used in therapy? I don't know, right? But we can anticipate. Dr. Harding can anticipate on this and jump in as well. So maybe it ultimately depends on how well it works, right? We saw pretty significant reductions in phenylalanine levels in the animal model with the original version. So our goal is to have a significant reduction in phenylalanine levels when you consume this bacterium. But we'll see when we ultimately get into trials. Maybe it's used along with medical foods as to help liberalize the diet, sort of like Kuvan is. I don't know that I envision medical foods going away anytime soon. I think a lot of the products we're working on now will be used in conjunction with medical foods. Maybe it's an alternative to Kuvan and PEG-PAL for those that aren't responders or aren't responding to PEG-PAL. It's great that we have PEG-PAL and Kuvan available, but it's not going to work 100% of the time for everybody. Maybe ultimately what we end up with is a sort of a cocktail of medicines that we use. We use medical food, and we use some Kuvan, and we use some of this probiotic product. That may be the direction that we're ultimately heading, and this is the direction that we see lots of fields of medicine. If you look at the treatment of leukemia today, it's not one drug that's used. It's a cocktail of drugs that's used for that do different things. What's interesting about this concept is I'm trying to attack the phenylalanine in the GI tract, right? Some of the phenylalanine could, in theory, come out of the body and be taken up in the GI tract, but it's a different mechanism than PEG-PAL, which is sort of attacking it once I'm in the bloodstream, right? So maybe there's an argument that says that I use both of them together, or you use a combination. But what we want I think as a community is we want to have a lot of different drugs available. You want to have a lot of drugs in development. I think it's great that there's 3 companies working on the same target. I think, God bless our friends at BioMarin, they did a lot of great work, but for a long time they were kind of the only drug company that were out there doing a lot of work in this field. So now you've got multiple companies, which is exactly what we want to see. This is, as I said, it's very risky. You don't know which one's going to work, so you want to have a lot of shots on goal, right? And then ultimately we want a lot of drugs available because what works for you might not work for you, might not work for you. So anyway, that's what we got. So we're very happy to be moving this forward, and we'll see how it goes.